molecule contributes a different required gene, then the cell will have all the enzymes required to synthesize tryptophan. Notice that complementation does not involve recombination. To do this test, the mutations must be in trans. If one molecule has both mutations, the other a wild type, and should be sufficient itself to confer a wild type phenotype unless one of the mutations is dominant. Therefore, having the mutations in cis serves as a control. The Cistron. This type of complementation test, called the cis-trans test, is used to define whether two mutations are in the same genetic functional unit. The genetic unit defined by the cis-trans test is sometimes called the cistron, a term essentially equivalent to a gene. As noted, two mutations in the same cistron cannot complement each other, and so when complementation is found to exist, this implies that the two mutations lie in different cistrons, that is, different genes. The term cistron is now rarely used to describing whether an mRNA has genetic information from one gene, monocistronic mRNA, or from more than one gene, polycistronic mRNA. Transposons and insertion sequences. The order of genes on the bacteria chromosome can be determined by methods of gene transfer. We have considered just as genes in eukaryotic chromosomes can be mapped by mating experiments. However, the exact arrangements of the genes along the chromosome is not necessarily permanently fixed. Some genes are capable of moving under certain conditions. The process by which a gene moves from one place to another in the genome is called transposition, and is an important process in evolution and in genetic analysis. We should emphasize that transposition typically is a rare event, occurring at frequencies to 10 to the minus 7, 10 to the minus 7, 10 to the minus 5, minus 10 to the minus 7 power per generation. Thus, the genes of living organisms are relatively stable. In addition, not all genes are capable of transposition. Rather, transposition of genes is linked to a presence of specific genetic elements called transposable elements. Transposition was originally discovered in corn maize, and then later in bacteria, owing to the extremely sensitive types of genetic analysis available in these organisms. It has now been shown that DNA sequences with the properties of transposable elements are widely spread in nature. However, in organisms with poorly characterized genetic systems, it can be difficult to detect transposition because it is a rare event. An event actually proves that a sequence is a transposable element. Transposable elements, as we discussed earlier. There are three types of transposable elements in bacteria: insertion sequences, transposons, and some special viruses, such as mu. In this section, we shall confine our discussion primarily to insertion sequences and transposons. Both these elements have two important features in common. They both carry gene encoding and transpose the enzyme necessary for transposition. Both have short inverted terminal repeats at the ends of their DNA. Remember that these ends are continuous with whatever DNA the element is inserted into. These repeats range in length from fewer than 20 base pairs in simple IS elements to more than 1,000 base pairs in some transposons. Each IS has a specific number of base pairs in its terminal split repeats. Such inverted terminal repeats are involved in the transposition process. Shows a genetic map of an insertion element named IS2 and a transposon named TN5. Insertion sequences are the simplest type of transposable element and carry no genetic information other than that required for them to move to new locations. Insertion sequences are short segments of DNA, about 1,000 nucleotides long, that can become integrated at specific sites on the genome. Insertion sequences, abbreviated IS, are found in both chromosomal and plasmid DNA, as well as certain bacteriophages. Several hundred distinct elements have been characterized, and most are designated by a number identifying its type. Because so many IS elements have been discovered, some receive designations related to the organism in which they were first identified. For example, is from Mycobacterium tuberculosis. IS elements are scattered about the chromosome, and the strains vary in the number and frequency of these elements. For instance, one strain of Escherichia coli has 5 copies of IS2 and 5 copies of IS3. Many plasmids also carry these insertion sequences, and it is homologous recombination between identical sequences on the F plasmid and the chromosome, not transposition, that allows the F plasmids to integrate into the bacteria chromosome and mobilize it. Some of the archaea have large number of IS elements in their chromosome. Transposons are larger than insertion sequences and carry other genes, some of them conferring important properties on the organism carrying them. These often include drug-resistant markers and other easily selectable genes. In addition, as we have mentioned, there are conjugative transposons. These transposons have genes allowing them to not only to move from one location on the bacteria genome to another, but also be transfer themselves from one bacterium to another. Some transposons are actually composite structures containing a gene or a group of genes lysing between 2 identical insertion sequences. The existence of such composite transposons indicate that new transposons probably continue to arise in cells that have insertion sequences. The mechanism of transposition. As mentioned previously, inverted repeats found at the ends of transposable elements are essential for transposition. The other essential component is the enzyme called transposase, which recognizes these repeats. This enzyme is usually encoded by the transposable element, although some very simple IS elements use an enzyme encoded by another genetic element. The transposase apparently recognizes, cuts, and eventually ligates the DNA during transposition. When a transposable element becomes inserted into another DNA, the target DNA has short sequences and the target DNA at the site of integration is duplicated. The target DNA sequences was not present in the element, but the transposable element has brought about a duplication of the DNA by the insertion process. The duplication of the target sequence arises because single-stranded breaks are generated by the transposase. The transposon is then attached to the single-stranded end that have been generated, and repair of the single-strand portions results in the duplication. Certain transposable elements prefer certain sequences as target sites, but others, including the bacteriophage mu, can insert themselves almost randomly for a representation of mu insertion. 2 mechanisms of transposition are known, called conservative and replicative. In conservative and transposition, such can occur in the transposon TN5, the transposable element is excised from one location in the chromosome and becomes reinserted at a second location. The copy number of a conservative transposon therefore remains at 1. By contrast, replicative transposons, such as bacteriophages mu, are duplicated and a new copy is inserted at another location. Thus, after a transposition event completed, one copy of the transposing element remains at the original site and another copy is found at the new site. During this whole transposition process, the source of transposition remains at its original site. At no time does the source transposon become free in the cell. Although the molecular details of transposition remain uncertain, and different transposable elements appear to have somewhat different mechanism models. For both conservative and replicative transposition are illustrated. As seen, single-stranded cuts are made at the ends of the transposon, at the sites of the inverted repeats, and staggered single-stranded cuts are made at the target site. The transposon is now joined to the target site via the single-stranded ends. In conservative transposition, the donor site is now cut, and replication repair then fills in the single-strand gaps in the target site. The process results in the formation of direct repeats in the target site at the ends of the transposon. In replicative transposition, the replication repair takes place, while the transposable elements is still attached to both original and the target sites. This leads to the formation of a composite structure called cointegrate. The final event in the pathway is resolution of the conjugative structure, leading to the release of original transposon and the presence of the new copy of the transposable boson at the target site. Transposition is essentially a recombination event, but one that does not occur between homologous sequences or use of general recombination system of the cell. It involves transposase, rather than the Rec-A protein that is involved in general recombination. Because this recombination involves a specific base sequence, it is called site-specific recombination. In contrast to homologous recombination discussed earlier in this chapter. Mutagenesis with transposable elements. If the insertion site for the transposable element is within a gene, insertion of the transposable boson will result in a loss of linear continuity of the gene, leading to mutation. Transposons thus provide a facile means of creating mutant throughout the chromosome. The most convenient element for transposon mutagenesis is one containing an antibiotic-resistant gene. Clones containing a transposon can then be selected by the isolation of antibiotic-resistant colonies. If the antibiotic-resistant clones are selected on rich medium on which the autotrophs can grow, they can be subsequently screened on minimal medium supplemented with various growth factors to determine if a growth factor is required. Transposons are also useful for incorporating an autotroph gene marker into the wild-type organism. Normally, autotroph recombinants cannot be isolated by positive selection, but if the autotroph marker to be introduced contain a transposable element with an antibiotic-resistant marker, then one can select for antibiotic-resistant clones, a positive selection procedure, and automatically obtain clones that have incorporated the autotroph marker. Two transposons widely used for mutagenesis are TN5, which confers neomycin and vancomycin resistance, and TN10, which contains a marker for tetracycline resistance. The bacteriophage mu is also widely used as a biological mutagen. Because mu integrates a wide variety of host sites, it can be used to induce mutations at many locations. Also, mu can be used to carry into the cell genes that have been derived